On the board up here, you can see part 5 of our genetics packet. It is the back cross / test cross. This is what I refer to as a test cross. Now, a test cross is something that we use when we do not know the genotype of a species. Okay? Now, when I go through and read this, it says, when an organism has the dominant phenotype, then its genotype can be either heterozygous or homozygous dominant. You can't tell by looking at it. In order to find out, we must use a test cross using a homozygous recessive organism. For example, we've been talking about brown eyes and blue eyes. Well, if something has brown eyes, it's either capital B, or capital B, lowercase b. If you and I are just looking at someone that has brown eyes, can we tell which one it is? No, we cannot. We cannot. So this is where we have to do a test cross. The example that we have here, it says, in Dalmatian dogs, the gene for black spot is dominant to the gene for liver-colored spots. If a breeder has a black spotted dog, how can she find out whether it's homozygous, capital B, or heterozygous, capital B, little b, spotted dog? If the breeder finds a black spotted dog whose ancestry is not known, she cannot tell it by looking at the dog whether it's capital B, or capital B, lowercase b. She should find a liver spotted dog whose genotype must be lowercase b, and mate it with the black spotted dog. This is the cross for a homozygous individual. So, what's happening? What's happening here, folks, is if we have a dominant that we don't know, whether it's heterozygous or homozygous, we use the recessive. Okay? We use the recessive. So, in this case right here, the recessive, okay, the recessive right here, if it's breeded with big B, is going to give me 4 out of 4. Oh, what are they? Black. I'm sorry. That's not erasing here, so we'll scribble it. 4 out of 4 black. So, if I have a dominant, a homozygous dominant, big B, it gives me every single puppy that those Dalmatians would have black spots. Okay? Now, if I had one that was big B, little b, can you see here that 1/2 are black, 1/2 are liver? Can you see that? So, if I mate those dogs and any of the puppies are liver, what does that mean? It was heterozygous, if any of them are. Because if it was homozygous, could they have a liver-colored puppy? No. Now, let's go back to probabilities. Could we mate these dogs right here? Could we mate these dogs right here and get 7 puppies, all of them were black? Could that happen? It could happen. So we could, there is the potential for a little bit of a false reading. Okay? A little bit of a false reading. Now, let's go to our example right here. And look at these. I know that these are all letter A, letter A, letter A. Okay? the auto setup must have been bad here. But it says, you found a wild black mouse. How would you determine the genotype of this mouse? It says, hint, white fur is recessive. Okay? So, what we're going to do is we're just going to start out by writing, okay, B, capital B is dominant for black fur. Lowercase b is the recessive for white fur, right? So, draw a Punnett square for possible crosses. Well, what we're going to do is I'm going to draw a square, and using a test cross, what do we always put on the side? We always put on the side the homozygous recessive. So I'm going to put little b here, big B here. So when I do my cross between these, all of these are going to have black fur, right? Here's my other one. It is big B, little b, and on the test cross, it's always little b's. Big B, little b, little b, little b, big B, little b, a little b, little b. So, it says you have 24 offspring. 23 with black fur, 1 with white fur. What was the genotype of the mouse you found? It's got to be this one, right? Because that's the only way we can get a white mouse, isn't it? So, that genotype of that mouse that you found had to be big B, little b. Now, here's the next question. If you had only 3 black offspring, can you tell what the genotype of that original mouse was? Okay? If you had only 3 black offspring, would you think that it would be one or the other? I mean, could it be either one? Okay? So, could you tell what the genotype was of that original one? No. You probably need more of a sample size, don't you? Okay? You probably need more of a sample size than 3 of them. And the whole reason is, based on our probability, folks, we know that if there's only 3 of them, and we have a 50% getting a black one right here, right? We have 50% chance of getting a black one right there. Well, that's just like a coin flip. Could I flip 3 times and get heads? Yeah. Yeah, based on probability, this does not tell us. What if it would say of the 3 offspring, or let's say that we only had 2 offspring, but one of them was white. Would we know then? Yeah, we would know then, wouldn't we? We would know then, okay? So that is, ladies and gentlemen, a test cross, okay?